We have time for a couple of questions. I'm sure there are some in the audience. If not, I have a couple. So we have a question up there.
So I have a technical question for Anna. So you did your experiment with 6 to 9 days organotypic cultures, right?
Yeah.
So the organotypic slices has an altered maturation with respect to the in vivo brain. So have you tried to perform your experiment at different ages or different days in cultures?
So actually the data for in that period of time is that the development in terms of synapse number, in terms of size, in terms of cell type in organotypic slices actually mirrors very well the developmental period of an in vivo brain. So if you did an acute slice at that time, you'd actually have very similar anatomy and physiology to the in vivo, to the organotypic slice. So actually we think that in this developmental period of time, the model works well.
So I'd like to ask a question to Irene. So my understanding in your presentation of your correlation between dopaminergic denervation and altered functional activity of the prefrontal cortex was that you were implying that the causal relationship was the dopamine to the frontal cortex function. So could you expand on this? Why you think the causal relationship, if I understood correctly, and why you think the causal direction is this?
Well, it's complex to say if it's the cause. Or
the reason for my question is that it seems that the main risk factor for impulse control disorders, if I'm remembering my literature correctly, is actually the dopaminergic drugs, right, and not the actual denervation.
Yeah, but apart from this, there are other factors involved, such as the neurobiology of Parkinson's disease and also the premorbid personality of people. A lot of Parkinson's disease in the same ranges of dopamine agonists, they didn't develop this behavioral complication. So we have seen that there is an association, but just that. I cannot say if it's first one thing or the other. Maybe it's a deregulation of all the system.
Okay, thank you.
Thank you.
In your work, you had Parkinson's patients and healthy controls. Have you thought of non-Parkinson's patients on dopaminergic drugs?
Yeah, it's a expected question. I think it's a limitation, in fact, of this study. I think with another group of non-ICD patients, most of all the metabolism will be more complete to compare these two groups. Yeah, I know.
So it seems we have another question.
To ask Marcelo. A very nice presentation, very good work. I would like to know if you have done the same experiments with Parkinsonian animals under levodopa therapy with L-DOPA in the two conditions, when levodopa is doing its therapeutic effects and also when it produces dyskinesias.
For the first condition, for your first the answer for the first experiment, for your first question, I'm sorry, is that experiment will be done next week, okay? So we're training this group of animals, and Eva is helping me, and we want to treat them with levodopa next week. We were not directly planning the dyskinesia experiment, but it will be an interesting thing to do also in this task, so we can talk later on that.
I'm afraid we're running out of time.
Given our change of schedule, we're going to have to stop a little bit earlier, and I really want to thank everyone for keeping this session on time. Thank you all for being here the last day in the morning, and we will start promptly our next session at 11:10. Okay.
Please be seated before 11:10.
Thank you very much.